Thank you all for coming today. I'm gonna let Ara introduce Dr. Loeb.
Thank you all for coming, both in person and on Zoom. Glad to have you. We have Dr. Loeb here to speak, and Dr. Loeb received her bachelor's in human biology from Stanford University, her master's and PhD in ecology from University of California, Davis. She has been a research ecologist with the U.S. Forest Service at the Southern Research Station located here at Clemson University since 1988, and is also an adjunct professor in this department at BC, also here at Clemson. For the past 25 years, she's been studying the ecology, conservation, and management of forest bats in the Southeast. Her research focuses on threatened, endangered, and sensitive species of the Southeast, the effects of disturbance on forest bats, bat responses to white nose syndrome, and developing and testing monitor methods for bats. Well, welcome, Dr. Loeb.
All right, well, thank you for the introduction, and thank you all for coming. It's as she said, I've been here since 1988. I came here on a 2-year postdoc and have been here ever since. And I often tell people, they ask me about my position, and I think I have probably one of the best positions. And I often tell people that I probably would not have stayed with the Forest Service had I not been located right here at Clemson. This has given me the opportunity to really have the best of both worlds, to work as a research scientist for the Forest Service in a mission-driven organization, but be in an academic position, situation, work closely with faculty and students. And so just want to take the opportunity to thank all of the people that I've had the opportunity to work with. Some 20 plus graduate students, some of whom I'm going to talk about today. And there is no way that I could have accomplished what I have over the last 36 years without the cooperation from people here at Clemson. So just want to take this opportunity to say thank you. And I guess he's not here, but also thank Eric. I think all graduate students who have worked with me since 2008 would second this, that they probably would not have been able to do their research quite as easily without the help of Eric Winters, both out in the field, GIS. So anyways, so all right, so 36 years with the Forest Service. I look at this career as basically providing science-based strategies for managing, conserving, and recovering species. These are some of the animals that I've worked on. Started off here working on southern flying squirrels and their interactions with great concated woodpeckers, and have been working on bats most recently. The endangered species that I'm talking about today is a tricolored bat, but actually, technically, it is not listed as endangered at this moment. It was proposed endangered in 2022. The decision was supposed to come out last September. Hopefully, it will come out by this September. I anticipate, as do most people in the bat community, that it will be listed either as threatened or endangered. And when we look at endangered species, and particularly endangered mammals, there are certain characteristics that make some species more likely to become endangered than others. They usually don't have all these characteristics, but usually check off a number of these. So oftentimes, particularly when it comes to mammals, large size, you know, the elephants, the rhinos, the big carnivores, tend to be a specialist, either a dietary specialist, habitat specialist, or both. Oftentimes have a small population size to begin with, and then some stressor comes along that pushes that population down. Smaller, limited geographic range, low reproductive rate, and oftentimes have economic or commercial value. When we look at bats worldwide, causes of endangerment are primarily habitat loss. Looking, loss due to harvesting of trees, conversion to agriculture, also hunting and collecting of animals, primarily in the tropics for the big tree bats. Disturbance is also a big factor, bats in caves, and urban development as well. So then we come to the tricolored bat, and want to give you an introduction to its natural history. So that you understand what I'm going to be talking about the rest of the seminar, but also to get an idea of where it fits into that paradigm. So first of all, this is a very small animal, 4.5 to usually 6 grams. Occasionally you'll have a really chunky female coming in at 7 grams. They have a large geographic range throughout the eastern United States, from Canada down into Mexico. They feed on a wide variety of arthropods. They're basically a generous insectivore, primarily a hawker, so catching insects on the wing. And we are finding now that they may be migrating over a long distance. They've been tracked with airplanes up to 250 kilometers, but some of the stable isotope analysis suggests that they may be migrating more than that. So that's something new. Given the fact that they have this large geographic range, you can find them in just about any ecosystem. So any place where there are trees for it to roost in, it can be found. And it can also be found I've caught them in parks in downtown Atlanta. So they're pretty adaptable, can be found just about anywhere. During the winter, in areas where there are caves, mines, extensive tunnel systems, they will use these for hibernation. So they'll go into these hibernacula, spend the winter there. They tend to go in earlier than most species and come out later. So go in about mid-October, early November, spend the winter in these hibernacula, emerge in late March to April. In areas where there are not underground hibernacula, in much of the southeast, they will use above ground hibernacula. So ridges, culverts, cavities in trees, and even canopies of some trees. So up in Spanish moss, cracks and crevices in canopies, of trees. So they're highly adaptable in the summer. They roost in live, primarily hardwood trees. They tend to like the bigger trees, but not always. They'll just hang up in a clump of dead leaves, usually a clump of hardwood leaves, but occasionally in a clump of dead pine needles. Also, we use moss, Spanish moss, particularly on the coast, lichens up in Canada. Tend to like trees that have lots of solar exposure, so more open areas that allows the females and the young to use that the solar exposure to thermoregulate. But in areas where it's warm, they'll use trees that have more canopy exposure. And during the active season, they will forage. They're small, so most people say, oh, they forage in interior forests, and they do, but they also tend to use open areas, and particularly the edges between open areas and forests. So again, highly adaptable. So when we go back to this paradigm in terms of what makes mammals endangered, how do pips, or I call them pips, fit into this? Large size? Definitely not. Dietary specialist, habitat specialist, nope. Small population size, possibly, but prior to their decline, we could catch them pretty much anywhere. Limited geographic range, low reproductive rate, yes. So like most temperate bats, they only have 1 litter per year, and they actually have 2 young per litter, which is actually higher than a lot of Myota species that only have 1. And they certainly don't have any economic or commercial value other than providing a lot of insect pest control. So why are they endangered? Well, white nose syndrome. For those of you not familiar with white nose syndrome, this is a fungal disease. It's caused by the fungus Pseudogymnoastis destructans, which I will refer to as PD. It is a psychrophilic fungus, so it likes cold temperatures. Grows anywhere between 1 and 19 degrees centigrade, but it grows fastest between 12.5 and 15.8. It invades the cutaneous tissue of the bat, particularly its wings. It will get on the nose, but its wings, its forearms, its legs, and it basically disrupts. It eats through the wings. I didn't show the ugly picture of a bat with holes in its wings, but it destroys the wings, and those wings are very important for maintaining physiological balance. So they tend to become dehydrated, their electrolyte balance is off, and also fungus causes them to arouse more frequently. And when they arouse from hibernation, they use up their fat reserves. So they basically are starving to death and dehydrated. The fungus can grow on them because when they are in that hibernacula, they are in that perfect range of the growth for that fungus. The other thing that makes this fungus very deadly is it does not require the bats to survive. So it can hang out in these hibernacula even when the bats are all dead, waiting for new bats to come in, and it's ready to invade new bats. So it was first found here in the US in 2006 in Albany, New York. By 2009, it was starting to creep north and then down south along the Appalachian chain. And by 2013, you can see that it started to come into the southeast, north part of South Carolina, north part of Georgia. And by 2018, it's pretty much covering a good portion of northern South Carolina as well as Georgia. And this is the most recent map, and you can see that is spread throughout much of the United States and a good portion of Canada as well. But what I do want you to notice is that there are parts of the Southeast, particularly the coastal regions, all of Florida, that are white nose free. So no fungus there, no disease there. Just to give you an idea of how these populations have declined. So if we look at the 3 on the left, these are the species that are most greatly affected by white nose syndrome, going to northern long-eared bat, the little brown bat, and the tricolored there in the middle. And so what we are looking at is basically close to 90 to 100% decline once these bats, the fungus gets in there. When we focus in on the tricolored bat, across its white nose is in 60% of the range, and within that 60%, we are looking at 93% declines in these populations. Hence the probable listing of the species. Just to bring this a little bit closer to home, and how this has affected tricolored bats around here. Black Diamond Tunnel is over Clayton, Georgia. It had probably one of the highest colonies of tricolored bat, hibernating colonies of tricolored bats, in the east, and, used to have over 5,000 bats. White nose comes in and, it declines a little bit the first year, and within 2 years, we are looking at 96% decline. So it went from 5,000 to less than 200 bats. Stumphouse Tunnel, which I will be talking about a lot more, originally had over 300 bats. White nose came in sometime in the winter of 2014-15, and we go in there February of 2015. Dead bats all on the ground, dead bats on the wall, and so it eventually went down by about 90%. And this is reflected in the summer populations as well, not surprising. So this is acoustic data from Georgia, and the green is where white nose is not, does not occur, and so populations are doing well. The yellow is where, we see the white nose, and again, activity in the summer is also considerably declined. So going back to 2013, this is when I actually got involved with white nose work, because it came here, and so gave me both the opportunity to work on it. And again, what we see is that a good portion of the southeast is not affected. So one of our first questions was, okay, how far south is this disease going to go? The fungus can get there, but are the bats going to actually be susceptible to the disease as it moves far south? So one of the first things we did was look at the torpor patterns of these bats. So for those of you not familiar with mammalian hibernation, just a quick recap, or if you physiology a little old, when animals go into hibernation, they drop their body temperature and keep it at a relatively low rate for several days, several weeks, but usually no more than about a month. And then they have a spontaneous arousal. And then for an hour, 2 hours, go back down into torpor, and then have another arousal. Do that throughout the winter. Their body temperature is usually about that of the environment, or maybe a degree or two higher. So again, to recap, PD grows between 1 and 19 degrees, but it grows fastest between about 10 and 15 degrees, or 16 degrees centigrade. So our question was, okay, these bats farther south, what are the hibernacular conditions? Do those hibernacula get down into that range? How far down do the bats go in terms of their body temperature? Does that get down into the range of the growth of this fungus? How long do they go into torpor? If they're only going into torpor for a short period of time and are aroused for the rest of the time, then they may be able to fight off that fungus, because not only does their body temperature drop, their metabolic rate drop, but their immune function drops as well. So these were the types of questions that we were interested in asking. And so we had a hibernacula where we knew that there was white nose, Stumphouse Tunnel, and we had a hibernacula down in Florida where we had tricolored bats. There was no fungus, there was no disease, and we compared the two to see whether these bats in Florida were going to be susceptible. So this is work that was done by Pallavi Sarajadan. And again, this is Stumphouse Tunnel up here in northwestern South Carolina. This is Old Indian Cave, in the panhandle of Florida. White nose positive, white nose negative. We put temperature sensitive transmitters, I lost my light, there we go, on these bats. They last about a month. And put antennas throughout the caves, and also put in low-tech data loggers that recorded the transmitter beeps. And so the warmer the bat is, the more frequent the beeps, we can translate that into body temperature. Okay, so this is just a sort of a typical trace of body temperature. And what we see is the bats doing the spontaneous arousal, but then going into torpor. Body temperature around here, around 15, 16 degrees. As we get towards March, when the bats are thinking about going out onto the landscape, we see that they are, those torpor bouts are getting much warmer. So what did we find? Well, first of all, we do find that the hibernacular temperatures in both South Carolina and Florida are well within that optimal growth range of the fungus. So even in Florida, it was actually even slightly colder. Also want you to notice the standard deviations here, because I'm going to compare that to some above ground roots, very, very stable temperatures, as well as very stable humidities, vapor pressure deficit. That's meaning that those are relative humidities close to 100% there. When we look at their torpor patterns, we see, first of all, that their skin temperature, again, right in that optimal zone for the growth of that fungus in both Florida and South Carolina. Their torpor bouts are relatively short compared to, say, a little brown bat, but they are going into torpor for about 3 to 4 days. And some of these bats are going in for quite long, up to 2 weeks. Arousal duration, they are arousing only for about an hour, hour and a half, so not very long. And they're coming up to close to normal thermal temperatures. So basically from this, we conclude that the environmental conditions in these Florida caves are certainly sufficient for the growth of this fungus. So if anybody takes down this fungus there, or bats take it down, it is quite likely that these bats will get white nose syndrome. Whether the winters there are long enough to see a lot of mortality, we don't know that, but it is likely that they will. I was really glad that we did not take the fungus down there. I was quite worried about that. So the environmental conditions are good. The bats are going into torpor long enough, and their skin temperatures are optimal. And these bats in Florida have very similar patterns to those in South Carolina. So we do conclude that, yep, they could be a bad sign for those bats in Florida. So that's below ground hibernacula. But I also mentioned that these bats in parts of the South where there are no below ground hibernacula are also using these above ground hibernacula. So how susceptible are they going to be to fungus? Do they go into a deep torpor? Is it long enough for the fungus to actually grow and invade them? And how long are they aroused? If they're active, they may be able to fight off that fungus. If they're feeding, they can restore their fat reserves. So this study we took on at Savannah Riverside, Blaise Newman was a student working with this. Those of you working at have ever been to Savannah Riverside, it's an interesting place. But one of the nice things is it's got a big fence, a lot of people with guns. So it keeps out people that you don't want, and you can put your equipment out on the forest and not worry about it. So we knew that there were tricolored bats using bridges there. So that's a nice, easy place to go and capture them, which is what we did. Use the same methods that Paula B did, put transmitters on the bats. But these bats were not just staying in the bridges. They were also moving. So we had to go track them to their trees. Once they got to the trees, we put the data loggers in front of the trees, and we're able to measure their body temperature or skin temperature as well. So again, looking at first environmental conditions, we see that roost temperatures are certainly sufficient for the growth of the fungus. But what we see is those standard errors. So yes, the mean temperature is within that range, but they are highly variable. And so the daily roost variation is about 5 degrees. So that can definitely play a part. The bridges are not quite that humid, but they're still pretty humid. Those cavities are again close to 100% humidity. When we look at the skin temperature of these bats, again, it's right there in that optimal zone for the fungus to grow. But again, we're seeing a lot of variation. So within a day, a bat's temperature could vary by about 5 degrees. So it could definitely go outside of that zone of growth. Torpor durations are shorter by about a day than they were in the underground hibernacula. But if you get a nice cold period, bats can still go into torpor for up to 2 weeks. When they arouse, that's about an hour longer than we saw in the hibernacula. And again, some of these bats were aroused for quite a long time. And the neat thing about the arousal was, and I didn't mention this for the bats in Stump House and in Florida, was that they were arousing at random times. So anytime during the day or night, they were arousing. But what we see here in these above ground roosts is that they are most likely to arouse just about dusk. So suggesting that they are sort of entrained to the environmental conditions and going out and foraging. So, and we did see pick up only a few pellets, but those pellets that we did pick up did have insect parts. So these bats are far more active than the bats in the 2 below ground hibernacula. They left the roots, the hibernacula or their hibernaculum roosts about a quarter of the nights, and they actually switch roosts about 13%. And not surprising, they're most likely to be active on one nights. So from this, we conclude that yes, these roosts do get cold enough to allow for the growth of the fungus, but it's highly variable from day to day and over the day. And they can go into deep torpor, but they don't always do that. Again, that temperature of the skin is varying over the day, and they're active on many nights. So their immune system may be kicking in. They tend to arouse during that normal active period, and they oftentimes will leave the roost. Therefore, we're hoping that these bats are far less susceptible to the fungus than are those in underground roosts. So we do know, I showed you that, yes, even in these hibernacula where you see, 90%, 95% decline, it's not 100%. And we see bats are persisting. So how are they persisting in these hibernacula? A couple hypotheses have been thrown out there. All bats have microbial communities on their wings, and it's possible that those microbial communities are changing, and some of those microbes may be taking, fighting off the fungus for the bats. They could be, their immune system may be adapting, and being able to better deal with the fungus. And the third hypothesis is that bats are changing their behavior and that they are starting to choose colder and drier sites within hibernacula or choosing colder hibernacula. And there's evidence for this in various places. And so we knew that the bats in Stump House were persisting, and but we didn't know why. And so we thought, well, maybe it is due to temperature. So we wanted to know whether they were changing, where they were roosting within the tunnel, and whether this was related to temperature, ambient conditions, and how this related to the change in their population. So for those of you who have not had the opportunity to go up to Stump House, if you're into mountain biking, that's now a big mountain biking park. But this is a tunnel. It's about 1,600 feet long, and it was supposed to go all the way. It was a railroad that they were going to build from Charleston up to, I think, Chattanooga. But when you go in there, and you can feel it any time of the year, but you will feel as you walk closer to it how cold it is in there. And it is certainly colder in the front part of the section, and as you go back, it gets warmer. So we've been censusing this population every year in late February. One time we did it 1st of March, but usually it's the last week of February. We go in there, we count every bat. We use a temperature gun to get its skin temperature as well as that of the wall. We measure the distance from the opening, and during 2020, 21 and 21, 22, we did this on a monthly basis. So this again just shows you that section A is considerably colder or somewhat colder than section B, and both of these are considerably colder than section C. And this pink area is that optimal growth range of PD, and you can see that most of the time section C is in that optimal growth range, whereas most of the time section B and section A and section B are below that growth range. So this is just to show you the change in the population. I showed you this before. We detected white nose. We detected the fungus in 2014. All the bats looked healthy. 2015 is when it looked really bad, all the dead bats. And it continued to decline to about 2018. And then we started to see this slight increase. And what we saw was that the bats changed where they were roosting within the hibernacula. So prior to white nose syndrome, almost all of the bats were in section C. Nice, warm, stable temperatures. As white nose came in, we saw fewer and fewer bats using section C, and by 2019 through 2021, significantly more bats using section A in particular, and somewhat section B. So we saw a significant change in where these bats were actually roosting. And that was not only the proportion of bats that changed, but the absolute number of bats increased in terms of who number using that section, cold section of the tunnel. And we also saw when Becky Brown looked at this at a monthly basis, that they were using that throughout the winter. So it wasn't that they were just moving at the very end of the season. And just to give you an update, that upward trend has continued. In 2023, we had 3 times the number of bats that we did at that nadir. So that this change in behavior seems to be working, that they are. And when we went in a week and a half ago, and there were 123 bats in there. So we are definitely seeing an increase. But here's the problem. So the bats prefer, or I won't say they prefer, but Section A is really important. It appears that Section A is important for them to, at least some of those bats, to use that cold temperature. Not all of the bats are using that cold temperature. Why not? Well, it could be because there are people going in and out of there all the time. And in general, they do tend to prefer those warmer temperatures. But the fact that Section A is important, and we've got people there, sets up this sort of conflict between recreation and conservation. Bats. This, as I mentioned, Stump House, over the last several years, they made it into this mountain bike park. And so visitation there is really growing. I don't think I've ever been there, and I'm there mostly during the middle of the week, at any type of weather, because it doesn't matter when we're in the tunnel, whether it's raining or not. There are people there. There are always people there. They come into the tunnel. They don't know what they're looking at. They have no idea that there are bats in there. They make a lot of noise because they like to make noise and hear the echo. So we were concerned about this. Increased disturbance to these bats that are already dealing with a disease. We've got white nose in there. People can take those spores out and transfer them to other places. And we started this study in 2020. There was a pandemic going on, and at that point, we didn't know about the connection between bats and COVID. So we were concerned about people going in there and transferring the virus to the bats. Whether they were susceptible or not, we didn't know to the actual disease, but they could at least become a reservoir. So what we did was set up a human dimension study. This was done with Sherry and Becky Brown, and looked at, first of all, knowledge of and attitudes towards bats, perception of conflict with bats, and in particular, what kind of support or what kind of things could we do to manage the interaction between people and bats at Stump House that people would tolerate. That's what we were really getting at. And Becky did some other things to just try to predict what people's very attitudes towards bats and how we would predict that. So did sort of, I guess, standard human dimensions kind of study, sent out mail surveys to 3,000 households in Cody, Pickens, and Raven counties. So people who might be familiar with Stump House. We had a pretty good response rate. We also did intercept surveys at the tunnel. So people went in, they came out, Becky asked them to fill out the survey when they came out. So they were definitely familiar with the tunnel. And just give you an idea about the demographics, not surprising, primarily white, mostly a little bit more than half male, And many were, again, not surprising, recreationists. Even those from the male survey oftentimes reported that they hiked and walked, and did mountain biking. And relatively highly educated, greater than 50% had at least a bachelor's degree. So surprising, at least to me, was that people are relatively educated about bats. At least they know that not all bats have rabies. They know a little bit about bats and that they control insects. They not so good on taxonomy. A lot of them think that bats are a rodent. But overall, pretty good. And at least, again, this was done within the COVID era, not that terrified that bats were going to give them COVID. In terms of attitudes towards bats, again, I thought pretty encouraging. Most people thought that they were valuable, beneficial, interesting. I was a little disappointed that not more people thought that they were cute, but what can you say? In terms of, we asked them about their tolerance for restricting access to recreation sites, just in general, in terms for wildlife, not specifically for bats and not specifically at Stump House. And people are pretty tolerant in terms of restricting access to preserve the health of an area, to ensure wildlife survival, ensure public safety. When it comes to limiting disturbance to wildlife or preventing the spread of disease, they're a little less tolerant of restricting access. Then we asked them, what could we do at Stump House that you would, how would you feel if we did these various management activities at Stump House? So people were pretty good. Yeah, they would be willing to keep their voices down. They would be willing to go through a chute cleaning station when they came out. They would be willing to not take their dogs in. When it came to limiting the number of days that people were allowed to enter the tunnel, a little less tolerant of that. And not so tolerant of complete closure of the tunnel. But what I found really interesting was that they were more tolerant of closing the tunnel completely than they were of wearing a mask. So again, this is what happens when you do it in the middle of the pandemic. So basically, what does this mean? Well, we do have some options if we decide to go there, if Wahala, who actually manages this site, is willing to, is to ask people to keep their voices down when they go into the tunnel, not ask people to not, or tell people they cannot take their dogs in, and maybe set up a shoe plating station. Probably increasing, we do have one sign out there, it's way away from the tunnel. We might be able to do a much better job of education and getting people on board and more supportive of bat conservation. So then that leaves us with, how do we manage bats out on the landscape? I mentioned before, they use big hardwoods, not very specific. Pretty much anything that has dead leaves in it. Wow, that's pretty much a lot of the trees out there. And they'll even use cows if they have to. They forage in pretty much anywhere, and they like managed forests as well. So how do we manage bats on the landscape? Good question. I don't know. I do know that this was based on studies looking at forest management effects on bats, but I'm willing to bet that this, if I were to go through all the literature on habitat use of bats and when they were conducted, again, most of them are conducted during the summer. People usually justify it because that's the maternity season, but that's also a great time for master's students to go out and conduct their studies. So it's probably a good reason why a lot are done in the summer. We know very little about actual habitat use during winter, spring, and fall. But the bats are out there active on the landscape during these times as well. And we all know that habitat changes considerably over the year. What forest looks like now is very different from what it's going to look like in the middle of July. And so bats may perceive these habitats very differently depending on the season. Obviously, the insect availability is going to be very different during these seasons. So we've looked at this some, we definitely need to look at this more. This is some work done by Kyle Shute down on the coast. We have very healthy populations of tricolored bats on the coast. That may be where the majority of the species will be in the future. So I don't have a graph for summer habitat use because the bats didn't care. They were just as likely to be in bottomland, hardwood forest, fields, ponds, salt marsh, upland. They showed no selection. But come winter, they did show selection. They were far more likely to use bottomland, hardwood forests than salt marshes and upland forests, and those fields and ponds. They were using them, but not quite as much. So this is showing that basically what we conclude from studies in summer may not apply to what goes on during winter. And I'm not going to steal Eduardo's thunder here. He hopefully will be down here in about a month to defend his thesis, but he looked at these bats up on the Endangered Pickens district and what are they doing in spring and fall. We saw not too many differences between spring and fall and summer. But again, it just shows that they tend to like open types of forests. So how do we recover tricolored bats? I'm not sure. And particularly when foresters come to me, I work for the Forest Service, so they think I know how to manage forests for bats. When it comes to how we manage forests for bat habitat, tricolored bats in the summer, spring and fall, I think it's going to be very difficult to give these foresters prescriptions. For an Indiana bat, I can say make sure that you keep X number of snags and maintain a good level of snags. It's really hard to say, make sure you have X number of big hardwood trees out there. So I think what may be important is looking at this at a landscape scale. How much forest is out there? How much can we maintain? The diversity of forests out there in terms of age structure and forest structure itself, so open forest, forest interior, and just maintaining that on a landscape scale. Winter habitat is a little bit easier to make some recommendations. These are underground habitats, the underground hibernacula. We know that having that ability to move from cold to hot, warmer, or from warmer to colder may be important in terms of surviving white nose. So concentrating on protecting those hibernacula that have that range of temperatures may be really important. And also working with the public in those hibernacula that have tourism There are many show caves out there, making sure that we can work with the public to decrease the amount of disturbance and transfer of white nose to other areas and transfer of our diseases to the bats. In unaffected areas, have a better understanding of how bats are using the landscape throughout the year. And also protecting these above ground hibernacula. So culverts and bridges, a lot of states are working with their DOTs now to conserve or basically protect these hibernacula. These striped-covered bats are not using them very much during the summer. So having more construction work done during the summer as opposed to during the winter. And hopefully South Carolina will get on board because they're going to have to in this species. So with that, I will answer any questions if there are any.
First off, that was great talk, but I was wondering, are there differences or do we know if there are differences in the microbial communities on the bat's wings between above ground and below ground hibernacula? And could that affect their PD susceptibility?
It could, oh gosh, there was a student at Kennesaw State that looked at that. And there weren't great differences, but it definitely could be. And people do see that there have been changes in the microbial communities once white nose has come in. But and you can take this, you know, some of these in the lab, you can kill PD very easily. And by taking some of these microbes and putting them in a jar, it will kill the colony of PD. But out in the field, it's hard for that to work as well. But yeah, good question. Thank you. Yeah.
The shift that you're seeing in Stumphouse Tunnel, it sounds like, so is that both individual behavior shifting as well as sort of natural selection acting on the population?
Well, so another thing that Becky did was put little tags on bats, fluorescent tags, and then we could measure from month to month. And some of the bats were moving forward, but not that much forward. So not really to account for that. So what we think might have happened is that those bats that use the front, that they might be coming back, they're surviving better, so they're coming back to the front. And that's why we wanted to look to see whether bats were moving. You know, the hypothesis was that as the fungal colonies grew on them, that they would then move, but it appears that they're not, they are staying there. So yeah, it's probably that those that use the colder temperatures are doing better. But we are seeing more and more bats in the back, so that there may be something else going on that these bats are being able to adapt. But there's still some bats in there that are not looking good right now.
Just curious your thoughts on how climate change might affect bats and PD. It seems like, for example, in Stumphouse Tunnel, it might increase the temperatures in like zone A and put them more in the range of susceptibility for PD to grow, but also in the, you know, in the hibernacula, like in trees that aren't underground, it might also cause those bats to become more active and wake up more frequently. So it seems like it could be both bad and good.
Yeah, I took that slide out. I did have a slide in here, and in the interest of time, I took it out. But yeah, what I saw was that I looked at February temperature and the wall temperature that they were looking at, that they were roosting at. And there is a positive relationship there, particularly in A. So I did propose that climate change could be a problem, particularly here in the southeast where they're sort of at that edge. So yeah, I agree, but then you have the above ground ones. So and basically there's a zone, you've got those areas that have the below ground hibernaculum where bats mostly are, and then you've got the other areas. Although Florida is a really interesting place. But you do have a lot of caves in Florida, but you also have bats using above ground hibernacula just in the same area. So they are starting to do some torpor studies down there, and looking at what is going on there, could be really interesting. Yeah. So.
Right.
Yeah.